Hey guys, today we're going to be discussing chapter 7 from the textbook, The Structure of the Nervous System. And so this is a really exciting chapter where we get to talk about the development of the nervous system from conception through gestation and on to adulthood. Now let's talk about the mammalian brain. So, for example, there are several different types of mammals here. But the human brain is by far the largest and the most complex. Remember that we would have an extremely large surface area if it wasn't for our sulci and gyri. These grooves and folds help to compartmentalize tissue so that we don't have to carry around extremely large brains. So the sulci and gyri essentially fold the tissue and to make it more compact. You can see that some other animals, like cats, don't have as much sulci and gyri, and rats and rabbits hardly have any as well. The smooth surface of the rat is important to note that it does not have sulci and gyri because it cannot perform as complex of cognitive function compared to the human brain. Additionally, the dolphin brain is very large, but brain size does not always equal better cognition. And we'll talk about dolphins later on in this class. Now let's talk about anatomical references. It's important to note that you are going to need to utilize anatomical references for the rest of this course or maybe even in your future career. This is a way that we can assign a specific part of the brain or body based on where it's located. The direction or anatomical reference that's pointing towards the rat's nose at the front of the brain here is known as anterior or rostral. This comes from the Latin word for beak. Interestingly, this direction at the back of the body, where the rat's tail is, or the back of the brain, is referred to as posterior or caudal, which comes from the Latin word for tail. Interestingly, it will be easy to remember that the top side is the dorsal side. You can think of the dorsal fin of a dolphin or a shark, whereas the ventral side comes from the Latin word for belly, so it's the underside. These terms, anterior, posterior, dorsal, and ventral, will be utilized to talk about the different areas of the brain, but they can also be used to describe different anatomical areas of the body in general. Interestingly, the right side of the brain and spinal cord are perfectly mirrored, and this is known as bilateral symmetry. So essentially, each brain region has two parts that are connected by both hemispheres together. We could say that we have one whole hippocampus, but we technically have two hippocampi, one in each sphere. Another way that we can describe the location of a different area of the brain is through this invisible line that's running down the nervous system called the midline. Whenever a brain structure is closer to the midline, it's referred to as a medial structure, whereas a brain structure that's farther away from the midline is referred to as a lateral structure. There are also different anatomical planes of section. For example, in figure 7.3, this is a rat brain. If you were to cut this brain right down the middle, down the longitudinal fissure, which would divide the left and right hemisphere from one another, this would be known as a sagittal section. The splitting of the brain into two equal right and left pieces is also referred to as mid-sagittal. Second, you could cut the brain in a different way in which both hemispheres would be in the section. This is known as a coronal cut. It's cut perpendicularly to the ground, and I think an easy way to remember this is that a coronal section is like a crown. It's going to provide us a section with both hemispheres. Now, if you remember back to chapter 1 and 2, we talked a bit about histology and how scientists use microtomes to take thin slices of brain sections. In an experimental psychology lab, you could take a rat brain and place it in an agarose mold, and you could take small sections of the brain tissue. And you could either take sagittal sections, which would only show you one hemisphere, or you could do coronal sections that would show you both hemispheres. However, it's important to note for a coronal section, if you cut too far in the back, you may only see brain regions that are in the posterior area of the brain. Similarly, if you cut right here, you could only see brain regions that are in the anterior portion or front portion of the brain. I'd also like to note that this right here in the front of the brain is referred to as the olfactory bulbs. Humans and rats have olfactory bulbs, and it plays an important role in olfaction. Later on, we'll talk about the olfactory bulbs in more detail, but it's interesting to note that this is one of the key sites for neurogenesis in adulthood, meaning this area of the brain allows us to create new neurons throughout our life. And this makes sense because a sense of smell and olfaction can be strongly associated with memory consolidation. Here's a better look at a coronal section. As I noted before, a coronal section allows us to see both hemispheres of the brain. So for example, if you cut right here, this would be cutting towards the more posterior area of the brain, and we wouldn't be able to see these important anterior brain structures. However, if we just wanted to look at the hippocampus, which is located here in the medial temporal lobe, we would be able to see that if we cut right here in this more posterior area of the brain. And as I noted earlier, we have bilateral symmetry of the brain, meaning we have two of each structure. We have one hippocampi in our left hemisphere and another hippocampi in the right hemisphere. And these come together to form the entire hippocampus. As you can see in a coronal section, we're able to see a hippocampi here in this hemisphere and in the right hemisphere. So ultimately, both hippocampi that come together will develop into one hippocampus. A pro of using the coronal section is that we're able to see both sides of the brain, whereas in a sagittal section, we would only be able to see one hippocampi rather than the entire hippocampus. Now we're going to introduce the nervous system. And the nervous system is made up of multiple subsystems, but the two main systems are the central nervous system, which is composed of the brain and the spinal cord, and the peripheral nervous system that's made up of the cranial nerves and the spinal nerves. We actually have 31 pairs of spinal nerves that are connected to the spinal cord, and then they go on to innervate other areas of the body, such as our muscles or our organs. We're not going to learn all 31 pairs of the spinal nerves in this class, so don't worry, but we will talk about how they function. If you go on Canvas, you have access to this worksheet I created. This is essentially a map that I created for you guys, so you can look at the connection between the central nervous system, the spinal cord, and the peripheral nervous system. The spinal cord ultimately is the main route of connection between the peripheral nervous system and the central nervous system. And throughout this lecture, we're going to talk about all of these different pathways and how information can be transported out of the brain or transported into the brain. But first today, we're going to talk about the central nervous system, and we're going to talk about how the central nervous system is composed of the brain, the cerebellum, and the spinal cord. For this example, I'm going to utilize a rat brain to describe the different parts of the brain. Hopefully some of this should be review because originally we did talk about the cerebrum and cerebellum when I talked about Galen's work with sheep brain dissections. And so here, these two hemispheres are referred to as the cerebrum, and behind the cerebrum is the cerebellum. If you remember, the cerebellum is home to Purkinje cells, which is a prominent neuron that has those beautiful dendritic branches. If you remember, the cerebellum can be referred to as the arbor vitae or the tree of life, which we'll look at towards the end of today's lecture. And then, of course, the spinal cord is connected to the brainstem. Also, this area right here is referred to as the sagittal fissure, which divides these two hemispheres. It's interesting to note that in general, the right cerebral hemisphere receives sensations from and controls movement of the left side of the body, whereas the left cerebral hemisphere here is concerned with sensations and movements on the right side of the body. This is known as contralateral control. Now let's focus on the cerebellum. So remember, the cerebellum lies behind the cerebrum, And although it's dwarfed by the large size of the cerebrum, it actually contains as many neurons as both of these cerebral hemispheres combined. And the cerebellum is our movement control center. It has extensive connections and axons that extend into the cerebrum and the spinal cord. And in contrast to the cerebrum, the cerebellum does not have contralateral control of the body. So the left side of the cerebellum controls movement on the left side of the body, whereas the right side of the cerebellum controls movement on the right side of the body. Now, the brainstem is one of the most important areas of the brain because it controls a lot of our vital function. The brainstem has this complex nerve group and fibers that extend to other areas of the brain to relay information from the cerebrum down to the spinal cord and from the spinal cord to the cerebrum. Interestingly, the brainstem has different areas such as the pons and the medulla oblongata, which help regulate vital functions like breathing, consciousness, and even the control of body temperature. One could survive damage to the cerebrum and the cerebellum, but damage to the brainstem is usually fatal because it controls such vital functions. Now here's a look at the spinal cord, which luckily should be review. The spinal cord is encased in this bony vertebral column and it's connected to the brainstem. So remember, the brainstem is kind of that pathway from the spinal cord up to the cerebrum and vice versa. So essentially, we've already seen a lot of these different things before because we've talked about Bell and Magendi's law. And we talked about how Bell and Magendi discovered the function of the dorsal and ventral roots. Remember that the dorsal and ventral roots innervate the spinal cord and their axons can go and project onto other neurons throughout the body or the brain. Interestingly, the dorsal roots receive sensation from the rest of the body, whereas the ventral roots can help command movement onto motor neurons at the neuromuscular junction or skeletal muscle. And now let's look at the peripheral nervous system. This is everything that's connected to the brain and the spinal cord and other areas of the body, like our cranial nerves and our spinal nerves. So now that we've talked about the central nervous system, we're going to focus on the peripheral nervous system. There are two different subtypes of the peripheral nervous system, known as the autonomic nervous system and the somatic nervous system. The autonomic nervous system innervates our heart muscle, our smooth muscle, and our glands. Some examples of this could be our gastrointestinal tract. Whereas the somatic nervous system innervates our voluntary skeletal muscles. The somatic nervous system is super crucial for voluntary control. All of the spinal nerves that innervate our skin, our joints, and our muscles are under voluntary control. Activation of the somatic nervous system allows us to exercise and walk. In contrast, the autonomic nervous system plays a role in regulating our involuntary control. It actually measures blood pressure and oxygen content in our arteries. And we have motor fibers from the autonomic nervous system that command muscle contraction and relaxation of our smooth muscles. For example, it can guide digestion in the intestines. It can help us regulate blood in the blood vessels and our cardiac muscles, such as the smooth muscle at the heart. Remember that when one speaks of an emotional reaction that is beyond voluntary control, like butterflies in your stomach, as shown here, it's usually mediated by the autonomic nervous system. This nervous system is also sometimes called the visceral nervous system, but for the context of this class, we're going to call it the autonomic nervous system. The autonomic nervous system is further broken down into two separate parts, known as the parasympathetic nervous system and the sympathetic nervous system. The sympathetic nervous system regulates our fight or flight response. And when the system is activated, it increases energy expenditure by increasing our heart rate, our blood flow, our blood pressure, and it uses a lot of the body's energy source. And that key energy source is glucose. So any glucose that has been stored in our muscle or our fat is quickly shuttled into the bloodstream and utilized during the sympathetic nervous response. Now, the sympathetic nervous system is typically activated when we're undergoing stress. This could be physiological or even psychological. When we get to the stress chapters of this class, we're going to talk about how chronic psychological stress can actually induce the sympathetic nervous system response. And that's not very healthy for us as humans because we were only built to really undergo this sympathetic nervous response when we're undergoing a physiological stressor. A good example of the sympathetic nervous response would be if you were walking down a dark alley by yourself and you felt like you were being followed. During this time, your heart rate would increase, your blood pressure would rise, and you'd have a sharp increase in glucose because your body is preparing to flee that dangerous situation. Conversely, the other subtype of the autonomic nervous system is the parasympathetic nervous response, and this is our rest and digest time, essentially the body's homeostasis. During this time, we can rest and we can slow down and reduce the amount of energy we're expending. During this time, we don't utilize as much glucose. Also, a great example of the parasympathetic nervous response is acetylcholine release at the muscarinic acetylcholinergic receptor in the heart muscle. Remember in Otto Louis' frog heart experiment, when he stimulated the vagus nerve of one frog heart, it increased acetylcholine release and helped slow down the heart rate. Interestingly, this is due to this parasympathetic nervous response. Also, this system aids in digestion. So for example, after you eat a large meal, your body is going to produce insulin that's going to shuttle glucose where it needs to go for long-term storage. We can store glucose and fat in our white adipose tissue in the form of triglycerides, and we can also store that in our muscle. Thus, the body is preparing for the sympathetic nervous response in the future by storing energy where it needs to go, so that when the sympathetic nervous system is activated, glucose and other energy sources can be utilized. So how is the central nervous system connected to the peripheral nervous system? Of course, we know the spinal cord is the main pathway for this, and this includes the spinal nerves that innervate it, in addition to the cranial nerves. The spinal cord communicates with both the body and the periphery systems via these spinal nerves. And remember, there are 31 pairs of spinal nerves. This is shown here in the purple circle. Spinal nerves exit the spinal cord through these notches between each vertebrae of the vertebral column. And each spinal nerve attaches to the spinal cord by means of the two branches, which we're very familiar with by now, the dorsal root and the ventral root. As we've talked about before in class, we know that the ventral root can synapse onto motor neurons. The ventral roots allow our motor neurons to go down to peripheral areas of the body to help command muscle and motor movement. Their cell bodies from the ventral root live in the central nervous system, but their axons can project down to other areas of the peripheral nervous system. We know this based on the example I gave at the very beginning of this course with the simple reflex. Sensory neurons, however, have cell bodies that actually live right outside of the spinal cord. So as you can see right here in red, this is the dorsal root ganglia. Now the reason they're called ganglia is because their cell body actually lives outside of the spinal cord. We know the spinal cord is a part of the central nervous system, and because the ganglia don't live inside of the spinal cord and they live right outside of it, they're referred to as ganglia rather than nuclei. Ganglia is another term for neuronal cell bodies that are not in the central nervous system but exist in the periphery. So if they lived inside of the spinal cord, they'd be referred to as nuclei, not ganglia. So how is information carried between the central nervous system and the peripheral nervous system? Well, of course we know this is axons playing a key role here. And there are two different types of axons I'm going to talk about, including afferent and efferent axons. Essentially, these types of axons tell us in which direction information is being carried to or from. Afferent axons you can think of as carrying to. A great way to remember this is that they move away from the peripheral body to the brain. So for example, if you received a sensation in your foot, the sensory neurons and these axons on the sensory neurons would carry that information up to the brain. Whereas efferent axons exit. They emerge in the brain and they exit the brain. So this is carrying from the brain to the periphery of the body. So after the brain registers a sensation from the dorsal root ganglia, then it's going to send out these messages via motor neurons that exit the brain and command muscles to move in response to that sensation. We're going to be utilizing these two terms a lot, so make sure you know that afferent axons are going to be carrying information to the brain, away from the body, whereas efferent axons on neurons are going to emerge in the brain and exit the brain to send information such as motor muscle commands down to the periphery of the body. I also added these images so we could look through this a little more closely. So in this image, the blue root here is the dorsal root. We know that sensory neurons innervate the skin. The ventral root is shown here in red, and we know that the ventral root has motor neurons that can synapse onto skeletal muscles to command our voluntary movement. Interestingly, remember I noted earlier that the dorsal root cell body lives outside of the spinal cord, and that's why it's referred to as the dorsal root ganglion rather than a dorsal root nuclei. Now, information that's being carried from the skin is going to go up to the brain, and that's why it's called afferent neurons, because they're being carried away from the periphery of the body. For example, if you felt something hot, such as a hot pan on the stove, your skin would recognize this heat and send signals up to the brain regarding this sensation. After this is registered in the brain, motor neurons are going to command movement via the ventral root, and efferent motor neurons that are exiting the brain are going to be synapsing onto skeletal muscle in your hand to ensure that you move away from that hot object. Remember that the cell bodies of the ventral root are located in the spinal cord, and that's one key difference between the dorsal and ventral roots. So quickly, just to review one more time, afferent neurons are going to carry sensory information away from the periphery of the body to the central nervous system, whereas efferent neurons are going to exit the brain and go to the periphery of the body, usually to command motor movements. Now that we've covered all of the different nervous systems of the brain, make sure you review that worksheet that I made for you guys. It's my map that I've created for all of the nervous systems and how they're interconnected. And now we're going to talk about the lobes of the brain. So this is a part of our central nervous system. There are 4 lobes of the brain, including the frontal lobe, shown here in blue, the temporal lobe, shown here in orange, the occipital lobe in red, and the parietal lobe in green. These 4 lobes contain different brain areas that all have different functions, and we're going to learn about the structures in each lobe as we learn how they develop throughout gestation. The brain is very well protected. This image here actually shows a human brain with the skull removed, so it just shows you the meninges. The meninges are these protective coverings that provide extra protection to the brain underneath the skull. And so these 3 membranes are referred to as the dura mater, the arachnoid membrane, and the pia mater. Dura mater comes from a root word, tough mother, so it's a harder surface area. And the arachnoid membrane specifically is this space right here. And in the subarachnoid space, CSF can flow. We also have the pia mater, which referred to as the soft mother. So this is a softer layer of meninge. If you've heard of meningitis before, meningitis is an infection of the meninges. So you could see how painful that infection would be This can be viral or bacterial, and how this could induce inflammation in an area of the brain that should not be receiving inflammation. Now, if you remember, I've talked about how the brain is essentially tethered. About 98% of the brain's weight is taken up by CSF because the brain is essentially floating in this cerebral spinal fluid known as CSF. If you remember, I introduced this in chapter 1 when we were talking about Galen. Remember that Galen was performing sheep brain dissections and found these large holes in the brain. This led to his ventricular theory, in which he thought that damage to this area could be very detrimental. It is true that you wouldn't want to damage your ventricles, as they do hold cerebral spinal fluid. Here we can see the lateral ventricles sit in the middle of the brain, and the third ventricle sits right beneath them. There's also this fourth ventricle here that connects to a spinal canal. This canal allows cerebral spinal fluid to go into the brainstem and the spinal cord. So the ventricles house CSF in addition to the subarachnoid space. So there is plentiful CSF in the brain. So what happens if the ventricular system is impaired? In this example, if the flow of CSF from the choroid plexus through that ventricular system to the subarachnoid space is impaired, the fluid could back up and cause a swelling of the ventricles. This swelling is referred to as waterhead or hydrocephalus. This sometimes can happen in newborn babies. So essentially, this is a leakage of CSF. And this can usually be treated pretty quickly. A drainage tube will be utilized and introduce the peritoneal cavity, and the tube is inserted into the lateral ventricle through a hole in the skull. Now, remember that a baby's skull is very soft and it hasn't fully developed and formed. So when the head increases in size in response to this increased intracranial fluid, the brain is not going to receive as much damage. Often, this condition goes unnoticed until the size of the head reaches enormous proportions. In adults, hydrocephalus is far more serious. The skull cannot expand, and intracranial pressure increases as a result. You wouldn't want to have leakage of CSF in the adult brain. Soft tissue can be compressed, and it can lead to death if not treated properly. So thus, hydrocephalus is much easier to treat in a baby that has a soft skull that has not fully developed in comparison to an adult. Now, in adulthood, hydrocephalus could sometimes occur from a traumatic brain injury. At birth, of course, hydrocephalus is due to this leakage, potentially in the lateral ventricles. Now, I hope you guys really enjoy the next portion of this lecture because we're going to start to talk about the development of the nervous system, right from conception throughout gestation and how our different brain areas develop in adulthood. Interestingly, our entire nervous system develops from one flat sheet of cells. So here's an embryo that begins as this very flat disc with 3 different distinct layers of cells. This includes the endoderm, which is shown here in this yellow color, the mesoderm, shown here in red, and the ectoderm, shown here in blue. The ectoderm is really important, and we'll see how it helps develop the nervous system. At this early stage, this is only at about 17 days from conception. The brain is simply this flat sheet of cells. So I want to note that in these images, the ectoderm is blue, and this is occurring around 17 days post-conception. So the walls of this neural groove right here are called neural folds. And the neural groove then forms into something called the neural tube. During this time, when the neural groove forms into the neural tube, this is referred to as neurulation. And I highlighted this because it's super important. This process called neurulation is the basis of the entire central nervous system. So every brain region, the spinal cord, all of these pertinent things in the central nervous system all develop from the walls of the neural tube. Interestingly, as the neural folds come together, some of this ectoderm here can be pinched off. This tissue is then referred to as the neural crest, and the neural crest develops into the entire peripheral nervous system. So this is all occurring around 17 days post-conception. So you can use this when you study, but essentially this is just a review of what I just showed you on the last slide. The neural groove becomes the neural tube, and through neurulation develops into the entire central nervous system, whereas the neural folds will develop into the neural crest, and when the neural crest pinches off some of that ectoderm, it develops into the entire peripheral nervous system. So remember, the central nervous system contains the brain, the brainstem, and the spinal cord, whereas the peripheral nervous system develops into the cranial and spinal nerves, all of those nerves that are going to innervate the skin and the muscle and other tissues and glands. So as I noted on my two previous slides, the process by which the neural plate becomes the neural tube is referred to as neurulation. And remember that during this process, this is the development of the entire central nervous system. It's so crazy to think that this happens 17 days into pregnancy because most women don't even realize they're pregnant at this time. Failure of the neural tube to close properly results in a fairly common birth defect that occurs in 1 out of every 500 births. 3 different types of neural tube defects we're going to talk about include anencephaly, spina bifida, and encephaloceles. These are 3 neural tube defects that result from failure of the neural tube to close properly. Remember that the fusion of the neural folds that form into this neural tube first begin in the middle at midline and then anteriorly and posteriorly. Failure of the anterior neural tube to close results in anencephaly. This is a condition in which there's degeneration of the forebrain. Unfortunately, due to this degeneration of the brain and skull, this defect is always fatal. Spina bifida is the failure of the posterior neural tube to close. And we're going to talk about different severities of spina bifida. Here's a quick look at both of these neural tube defects I just introduced. As you can see, anencephaly prevents the growth of the brain and the skull. Unfortunately, due to this, it is always fatal. Spina bifida has different forms of severity, but this is what it looks like, and it's characterized usually when a baby is born with large dark hairs on their back in the spine area. Interestingly, the word bifida comes from the Latin word meaning cleft in 2 parts. So there can be severe or less severe cases of spina bifida. Less severe forms are characterized by these defects in the meninges. As you can see here, there are vertebral arches missing, overlying this posterior area of the spinal cord. Doctors can usually note if a patient is born with spina bifida because the child will have long black hairs on this area of the spinal cord. This form of spina bifida is usually easy to treat with surgery. However, more severe cases of spina bifida resulting from the failure of the posterior spinal cord to form from the neural plate can be really dangerous. This is known as spina bifida cystica. As you can see, there's this large bulge in the back of the spine, in this posterior area specifically. And due to this, there could be meninges or cerebral spinal fluid, or even parts of the spinal cord that are growing outside of the spinal area. This is usually harder to perform surgery on, but sometimes there can be success rates with this. Encephalocele is a sac-like protrusion or projection on the back of the brain and the membranes that cover it through an opening in the skull. Encephalocele happens when the neural tube does not completely close during pregnancy. This can result in an opening anywhere along the center of the skull, from the nose to the back of the neck, but most often at the back of the head like it's pictured here. So, researchers estimate that about 1 in every 10,500 babies born in the US are born with encephalocele. It has a 55% survival rate, and the surgery can be a little bit difficult. There are different severities of encephaloceles similar to spina bifida. For example, here, maybe there's just CSF or dura. Remember those meninges that come outside of the back of the brain? But in more severe cases, parts of the brain or even ventricles from the brain can develop outside of the back of the brain and protrude out of the skull. This is treated with surgery to place this protruding part of the brain and the membranes, covering it back inside of the skull and then closing the skull. However, neurological problems caused by encephalocele will still be present, and long-term treatment can depend on each child's condition. Multiple surgeries are usually needed depending on the location, and this can be a really difficult treatment. So how can you prevent neural tube defects? Maternal nutrition plays a key role in successful neurulation, and that's because neural tube defects can be tracked to a deficiency of a vitamin called folic acid or folate. So this is occurring during the first few weeks of conception. We know that neurulation occurs about 17 days after conception. Most women don't even know they're pregnant during this time. So women who are expecting or thinking of trying to have a baby should try to take prenatal vitamins that provide plentiful folic acid. Folic acid plays an essential role in a number of metabolic pathways, including the biosynthesis of DNA. Studies have shown that maternal folic acid supplementation can reduce risk of neural tube defects by 90%. Women should be taking a multivitamin with at least 0.4 milligrams of folic acid before planning pregnancy. And good dietary sources of folic acid include leafy greens, yeast, eggs, beans, and oranges. A lot of the breakfast cereals we consume are also fortified with folic acid. But again, I recommend to any women in this class, it's super important to take 0.4 milligrams of folic acid, even if you're not planning on becoming pregnant, as it's so important to be taking this during that 17 days post-conception. The entire human brain is derived from 3 primary brain vesicles. So here again, we're very familiar with the neural tube. Those 3 brain vesicles include the prosencephalon, which will develop into the forebrain or the front of the human brain, the mesencephalon, which will be referred to as the midbrain, and the rhombencephalon, shown here in green, which will develop into the hindbrain. These 3 primary vesicles will end up becoming several different brain structures in adulthood. But this is what we'll refer to them as during gestation. This process in which these 3 primary brain vesicles start to develop in a baby is called differentiation. Remember that the prosencephalon is the forebrain, so this is the front of the brain, and it differentiates into 3 different substructures, including the telencephalic vesicles, the diencephalon, and the optic vesicles. As you can see here, we know that we have 2 hemispheres of the brain that create the cerebrum, and we can already see it starting to develop, even at this very early time in conception. These secondary structures that sprout off, including the telencephalic vesicles and the optic vesicles, are going to play a really important role in the development of future brain regions in the adult brain. Diencephalon is also referred to as between brain, and this makes sense as it's between both of these telencephalic vesicles. The diencephalon is actually going to end up becoming a structure known as the thalamus in the adult brain. And we're going to talk about how the thalamus plays an important role in communication within the brain from the cerebrum to the cerebellum and vice versa. The mesencephalon is here in red, and this is known as the midbrain, whereas the rhombencephalon in green is shown as the hindbrain. We're going to talk about different midbrain structures and different hindbrain structures that develop throughout differentiation and eventually become these structures in the adult brain. Now we're going to talk about the development of each primary brain vesicle, including the forebrain, midbrain, and hindbrain. I'm going to focus on each one separately and teach you about all of the different brain regions that are going to develop from each primary vesicle as the baby develops throughout gestation and then later on into childhood and adulthood. So the forebrain, also known as the prosencephalon, has 3 secondary vesicles that I've already introduced, including the optic vesicle, the thalamus, also referred to as the diencephalon, and the telencephalon. Remember that the telencephalon develops into 2 different bulbs, the telencephalic vesicles. Now, the optic vesicle, interestingly, derives into the retina and the optic nerve. So that is helpful to remember that the optic nerve develops from the optic vesicle and thus plays an important role in our vision. The thalamus is also referred to as the diencephalon, and this develops into the dorsal thalamus, the hypothalamus, and the third ventricle. Later on in chapter 15, I'll teach you all about the hypothalamus and the important role it plays in our endocrine system and our stress response. Now, the telencephalon breaks off into the cerebral cortex. This is important because the cerebral cortex is this large amount of surface area on the cerebrum. Additionally, the olfactory bulb, which I pointed out at the beginning of class, is in the very front of the brain and plays an important role in olfaction. We're also going to talk about these other brain structures and how they play an important role in communication between regions and hemispheres of the brain. So here are some forebrain structures you should know. The first is the cerebral cortex. Now remember, the cerebral cortex is this large surface area across the cerebrum, and essentially each area of the cortex may have a different function depending on where it's located. The hippocampus is in the medial temporal lobe of the brain and plays a vital role in the neural mechanisms of learning and memory. The amygdala also plays an important role in our processing of fear-based memories and strong emotions. The thalamus is a key relay station in the brain that helps communication from the cerebrum to other areas of the brain. And we'll talk about these important neurons known as the internal capsule that help relay this info. The hypothalamus, which we're going to talk about largely in chapter 15 because it plays a crucial role in endocrine function and homeostasis. The hypothalamus helps guide other glands and regions to produce neurohormones or sex hormones that are going to be secreted and dumped into the bloodstream. Also, the corpus callosum, which is this large commissure or bundle of axons that allows information to cross from each hemisphere to the next. The retina, which plays an important role in transducing our visual information in our eye. And the basal ganglia. The basal ganglia is a super important structure that helps us refine our motor movements and our motor planning. Basal ganglia injuries and neurological diseases that are associated with injury to the basal ganglia include Tourette's syndrome and Huntington's disease. Interestingly, the basal ganglia should really be referred to as the basal nuclei. Earlier, I told you guys that ganglion are usually only in the peripheral nervous system. This is the only example of a brain region that's referred to as the basal ganglia. Although these neurons live in the brain, they're referred to as ganglia. So ultimately, the basal ganglia should be referred to as the basal nuclei, even though it's located in the brain. So here are some of the structural features of the forebrain throughout the development of a child. So here, if we look right here in this area, this is the development of those 2 telencephalic vesicles, and below them is the diencephalon that will eventually develop into the thalamus and the hypothalamus. We know that the telencephalon will end up becoming the cerebral cortex, which is a really large area of tissue with different functions depending on the area. Also, the corpus callosum here, as I noted earlier, are these axons that are going to cross over from the left hemisphere to the right or from the right hemisphere to the left, and this allows communication between hemispheres. Also, the internal capsule is an important area that helps regulate communication between the cerebrum and the thalamus. Obviously, ventricle development plays an important role in our developing brain. Remember that the ventricles are these fluid-filled spaces filled with cerebral spinal fluid, or CSF. And so CSF can develop in the lateral ventricles, as shown here, and in the third ventricle. In the adult brain, whenever you see these paired lateral ventricles, you know this tissue that's surrounding them is the telencephalon. The space at the center of the diencephalon here is the third ventricle. And if you remember, the diencephalon here is going to end up being our thalamus in adulthood. So here are some other gray matter structures. If you remember at the very beginning of this class, I introduced gray matter versus white matter. Remember that gray matter is tissue where there are large groups of cell bodies or nuclei. This is also referred to as the soma of neurons. Whereas axons are referred to as white matter. Axons are these large extensions of neurons that can branch off to different areas of the brain. Now, the walls of the telencephalic vesicles appear swollen due to the proliferation of neurons in this area. And this proliferation of neurons helps us to create our gray matter structures, the cerebral cortex and the basal telencephalon. Now, the diencephalon I've already talked about develops into the thalamus and hypothalamus. The thalamus gets its name from the Greek word for inner chamber. It's in this inner part of the brain and helps with that communication from the cerebral cortex to other areas of the brain. And of course, we'll talk a lot about the hypothalamus. Again, I've noted that the hypothalamus plays a really important role in endocrine function and hormone production. Now let's talk about white matter structures. So I've already reviewed that gray matter consists of the cell bodies or the nuclei. The axons that project from those nuclei develop into bundles referred to as white matter structures. And these axon bundles together form three major white matter systems: the cortical white matter, the corpus callosum, and the internal capsule. Now, the corpus callosum, remember, is this large commissure in the brain that allows information to be passed from the left hemisphere to the right and vice versa. The internal capsule is this bundle of axons that allows for communication between the thalamus and other areas of the brain. For example, the internal capsule can regulate information from the cerebral cortex to the thalamus. So as I noted before, the internal capsule is a bundle of axons that help transduce information from the thalamus to the cerebral cortex. So really the thalamus is a gateway of sending information from the cerebral cortex to other areas. Information from our eyes, our ears, and our skin are brought to the cerebral cortex via the thalamus, and that's helpful thanks to the internal capsule that has long axons that allow information to be transduced from the thalamus to the cerebral cortex. Now, as I noted before, the hypothalamus helps us regulate homeostasis and neuroendocrine function and endocrine function in the body. And so the hypothalamus largely controls the autonomic nervous system response, which, if you remember, that's our involuntary responses. It helps regulate bodily functions in response to what we need. And as shown here in blue, this is our hypothalamus, right below the diencephalon or thalamus. And right below the hypothalamus, you can see this tiny little gland right here. This is the pituitary gland. The hypothalamus has specialized neurons that have long axons that project into the pituitary gland and help guide it to either produce or secrete certain types of hormones that we're going to talk about. Thus, it plays an important role in motivating us to find food, drink, and it also helps in the sex response. It also regulates our visceral fight or flight response, so in the sympathetic nervous system. And the hypothalamus ensures that the brain is well nourished by commanding the autonomic nervous system, which will increase peristalsis, like movement of material through the gastrointestinal tract, and it even redirects blood to the digestive system. Chapter 15 is largely going to talk about all of these different roles of the hypothalamus and how it can even play a role in producing hormones that make us thirsty to make sure that we drink when we need to. Ultimately, the hypothalamus is doing God's work, and it has a lot of different roles that we're going to talk about later on. So here's an image of a sheep brain, and this sheep brain was cut perfectly in half, and this would be a mid-sagittal cut or section of the brain so that you can see exactly one perfect hemisphere. When we look at a sheep brain, we can see first the spinal cord is here, and right in the hindbrain area, we have the medulla oblongata and the pons. These two brain areas play a really important role in vital functions like respiratory function. And right up here, we see our beautiful cerebellum. As I've talked about before, the cerebellum is home to plentiful Purkinje cells. Remember, those Purkinje neurons have really long dendritic branches. That's why this is referred to as the arbor vitae or tree of life. It's just so beautiful. And this is a great way for us to also see the difference between gray and white matter. So the gray matter or the darker color is the nuclei, whereas this white matter are axons and long dendritic branches that are coming off of those Purkinje neuron cell bodies. There are some other areas of the brain that we're going to talk about. We've already discussed the cerebrum, and in my earlier lectures, we actually talked about the pineal gland. If you remember, Descartes thought that the pineal gland was the seat of the soul because it was directly in the center of the brain, but this gland specifically creates and secretes melatonin for us. Also, the corpus callosum is right here. I've talked about the corpus callosum and how it's this large bundle of axons that helps send information from the left hemisphere to the right and vice versa. So this bundle of axons allows for communication between both brain hemispheres. Here we have our optic nerve, which we know plays an important role in our vision. The thalamus here, which is our relay center that helps transmit information from the cerebrum to other areas of the brain. And right below that is the hypothalamus, which plays a large role in endocrine function. Now, on your next exam, you will have to label a sagittal section of the brain, so make sure you refer to this. And I've also created a neuroanatomy worksheet for you guys that's posted on Canvas. I definitely recommend printing that out because the worksheet has practice pages in it where you can practice labeling different parts of the brain. And then here's another view of the brain to show you some other important structures. I've mentioned the amygdala before, which comes from the root word almond. It's kind of an almond shape, but it sits right beneath the hippocampus. So here's the hippocampus. The hippocampus is this large curved structure in the medial temporal lobe. And so we'll talk about it in a few slides. It kind of looks like a seahorse when you remove it from a brain. And then the thalamus sits right here in the middle. And remember that the thalamus can communicate with the cerebral cortex via the internal capsule axons. So again, you can utilize this to help you practice labeling the different parts of the brain. For your exam, I'm primarily going to be focused on the amygdala, the hippocampus, and the thalamus in this exact image. But when we look at sagittal sections of the brain, you're going to need to label several other areas like the brainstem and hindbrain structures and the cerebellum. But again, make sure you look at the neuroanatomy worksheet because that's going to help you determine what structures you should focus on while you're studying. As I mentioned before, the basal ganglia plays a vital role in fine motor movement and directing our muscles. So essentially, any type of disorder of the basal ganglia is going to affect a person's motor abilities. Huntington's disease is an example of this. You may have heard of Huntington's disease, but it is a fatal disease. It usually only affects 1 in every 10,000 Americans, so it's very rare, but it's caused by a dominant gene that codes for a very long protein called huntingtin. Huntingtin proteins can accumulate together and trigger neuronal cell death in the basal ganglia. The function of normal huntingtin protein is actually unknown still. Researchers are still trying to see what it does in a normal, healthy brain, but in a disease state, it can trigger this programmed cell death in the basal ganglia. So thus, basal ganglia disorders like Huntington's disease can affect motor movement and prevent motor neurons from doing their job in this involuntary muscle control. Here is an image of a fully dissected human hippocampus. As I mentioned before, the hippocampus is long and curved. So when you remove the hippocampus from the human brain, it actually resembles the shape of a seahorse. Now, as I've noted before, a lot of times neuroscientists have figured out the primary function of a certain brain region or area due to some type of accident or traumatic experience in a patient. And that was the case of patient HM. So patient HM, his name was Henry, and he lived during the 1950s. And in the 1950s, we still didn't know the true purpose and function of the hippocampus. But Henry was experiencing debilitating seizures from epilepsy, and so he went to a neurosurgeon to try to figure out how they could cure his epilepsy. Well, the neurosurgeon completely removed Henry's hippocampus. Here's a normal brain, and here's Henry's brain. He took out this entire hippocampal structure out of his medial temporal lobe. We understand now that the hippocampus plays a vital role in learning and memory because Henry developed anterograde amnesia. This means that Henry could no longer develop new memories. So this was very difficult for him. Interestingly, though, the surgery did help him with his epilepsy. So although he no longer had seizures, he could no longer develop new memories. So now this procedure would never be done because we understand the vital role the hippocampus plays in the brain. Interestingly, as I've noted before, Alzheimer's disease is the 7th leading cause of death in the United States, and it's a neurodegenerative disease that's characterized by these amyloid beta plaques and hyperphosphorylated neurofibrillary tau tangles that develop in key areas for the neural mechanisms of learning and memory. Dr. Alois Alzheimer discovered Alzheimer's disease in the early 1900s when he was working with a female patient. She was middle-aged and she was presenting signs of what we now know as dementia, but at the time he just thought it was severe memory loss and he didn't know what was causing this. She died very young, and during a postmortem autopsy of her brain, Dr. Alzheimer found that there was severe shrinkage of her brain. Here's a healthy brain on the left, and on the right, this is a brain that's severely shrunken from Alzheimer's disease. This shrinkage of the brain is known as brain atrophy. An atrophy of the brain in these key areas can result in dementia and the memory loss in dementia and Alzheimer's disease. So as you can see here, there's severe shrinkage or brain atrophy in the cerebral cortex, and then severe shrinkage of the hippocampus. And so thus, this is how Dr. Alzheimer discovered how important the hippocampus, again, played a vital role in learning and memory function for this patient he was working with. The retina is actually like a window to the brain because it's the only part of the central nervous system that you can see without an invasive procedure. You can look at the retina for signs of macular degeneration, diabetic eye disease, detachment of the retina, and degeneration of the rods and cones in the retina. Now, before I go on to discuss the midbrain structures, let's quickly review the forebrain, which is also known as the prosencephalon. Remember that the forebrain is broken up into subtypes, including the telencephalon and the diencephalon. So here in this image, we can see that the diencephalon is colored yellow. This is because the diencephalon contains the thalamus, the hypothalamus, and pineal gland in the adult brain. Other important adult regions that develop from the telencephalon include the cerebral cortex, the basal ganglia, the hippocampus, and the amygdala. Next, we're going to start talking about midbrain structures, which are also referred to as the mesencephalon. The midbrain or mesencephalon has several adult derivatives, including the tectum, the tegmentum, and the cerebral aqueduct. Let's talk about how these develop during differentiation of the midbrain. When the midbrain differentiates, the three primary brain vesicles are going to develop into adult derivatives. The midbrain, shown here in red, is going to develop into the adult derivatives known as the tectum, the cerebral aqueduct, and the tegmentum. The aqueduct connects rostrally with the third ventricle of the diencephalon, which of course the adult derivative of the diencephalon is the thalamus. So now let's look at some crucial midbrain structures, and I've color-coded them so it's easier for us to see them in this labeling. So as you can see in purple, this is showing us the tectum. The tectum is going to develop into these two structures in the adult brain known as the superior and inferior colliculus, and they play an important role with auditory and visual processing. The tegmentum, shown here in pink, helps us plan out coordination of movement and pain processing, and later we will talk about its adult derivatives. Both the superior and inferior colliculus are hidden by the cerebellum. Thus, the cerebellum was removed from this image so that we could see these important midbrain structures. The superior colliculus receives direct input from the eye and thus plays an important role in visual processes. It helps to control eye movement, which it does via synaptic connections with the motor neurons that innervate the eye muscles. Additionally, the inferior colliculus, which sits right beneath it, ventral to this area, receives sensory information from the ear and thus plays an important role in audition. The midbrain contains axons descending all the way from the cerebral cortex and down the brainstem to the spinal cord. Let's take a closer look at this area. As I noted before, if you pull back the cerebellum, you can actually see some of these structures. Here's the superior colliculus, and underneath it right here is the inferior colliculus. We're looking at a sheep brain right now, but it's also important to point out that this is the pineal gland right here. If you remember in the earlier chapters, in chapter 1, I talked about René Descartes and how he was a philosopher that he believed the pineal gland was the seat of the soul just because it was in the very center of the brain, when in reality we know that the pineal gland actually plays a crucial role in melatonin production now. Here's another midbrain structure known as the tegmentum. It contains both the substantia nigra, shown here in turquoise, and the red nucleus. Both of these areas play a really important role in our voluntary movement. There are other cell groups scattered in the midbrain that also help regulate consciousness, mood, pleasure, and pain. For example, the substantia nigra has a large amount of dopaminergic neurons that play a role in motor movement. The substantia nigra is actually a key part of the basal ganglia, and we're going to talk more about that on the next slide. The red nucleus shown here is a structure in the rostral midbrain that's involved in motor coordination. And the reason it's this pale pink color is it's believed to have the presence of iron in two different forms, including hemoglobin and ferritin. As I noted in my previous slide, the substantia nigra plays an important role in the basal ganglia system. It's a part of the basal ganglia, and degeneration of cells in the substantia nigra can lead to Parkinson's disease. These midbrain structures are small and often scattered nuclei. Unfortunately, patients with Parkinson's disease usually have lost more than 80% of the dopamine utilizing neurons in their substantia nigra. Does anyone remember the drug that is typically used to treat Parkinson's disease? We talked about this when we discussed neurotransmitter synthesis. If you remember, the precursor for dopamine is called L-DOPA. L-DOPA is a drug that's given to Parkinson's patients to help increase the production of dopamine levels due to this loss of dopamine-producing neurons in the substantia nigra. Overall, it's important to know that the basal ganglia plays a crucial role in voluntary motor movement. There are three key structures in the basal ganglia, including the substantia nigra, shown here, which we just talked about in the previous slide, the putamen, shown here in green, and the caudate nucleus, shown here in green. The caudate nucleus and the putamen are also known as the striatum. All of these structures, the substantia nigra, the caudate nucleus, and the putamen, collectively known as the striatum, play a crucial role in motor movement. Thus, any damage to these areas of the brain are usually associated with neurodegenerative diseases like Parkinson's disease. Damage to the midbrain and cerebellum can be really dangerous. For example, damage in these areas can lead to impaired balance and coordination. There is a degenerative disease known as ataxia that can affect someone's voluntary coordination of muscle movements. And any damage to these areas of the brain, such as the basal ganglia, can result in ataxia, in which a person may not be able to have normal coordinated movements. There are also other ataxia symptoms that you can look out for. Now we're going to talk about the development of the hindbrain, which is also known as the rhombencephalon. The rhombencephalon will end up developing into a few adult structures, including the cerebellum, the pons, the fourth ventricle, which holds cerebral spinal fluid, and the medulla oblongata. If we look at this first image here, this is a sheep brain. We're looking at the ventral view of the sheep brain, meaning it's flipped over so we can see its underbelly. The pons sits right here, and the medulla sits underneath it. We can also look at these structures here in a sagittal brain section. If you remember, this sagittal view is when the brain is cut in half perfectly so that we have two equal sizes of each hemisphere. We can already see structures we've reviewed previously. We've looked at forebrain structures like the thalamus and the hypothalamus. We've looked at midbrain structures such as the tegmentum and the tectum. If you remember, those are responsible for visual and auditory cues and motor movement. And then down below that, we have the pons and the medulla. Here's another image that I've shown before, but this is from a sheep brain. This is a sagittal section again of a sheep's brain. And as you can see, the spinal cord in the hindbrain is connected. Then we have the medulla oblongata, and we have the pons sitting right here. If you remember, I've talked about how these hindbrain structures are super crucial because they help us with vital functions like breathing and respiration. So thus, damage to the hindbrain specifically can be really dangerous. Now, the medulla oblongata is an amazing structure. The crossing of axons in the medulla explains why the cortex of one side of the brain controls movements on the opposite side of the body. If you remember, that is called contralateral control of the body. And here in this image, it's easier to see where the medulla oblongata sits in the hindbrain. Now, the pons is a hindbrain structure that plays a crucial role in communication between the cerebral cortex and the cerebellum. There are many nerve bundles that lie in the medulla and the pons, but about 20 million axons in the human brain synapse onto neurons in the pons specifically. And pontine cells relay all of this information to the cerebellum on the opposite side. And so thus the pons serves as a massive switchboard connecting the cerebral cortex to the cerebellum. Because if you remember, the cerebellum does not have contralateral control. The left side of the cerebellum controls motor movement on the left side of the body, whereas the right side of the cerebellum controls movement on the right side. This is different from the contralateral control that we see in the cerebrum specifically. So the pons is playing a really important role here as the switchboard between these two brain areas. The cerebellum receives massive axonal inputs from the spinal cord and the pons. It basically serves as this gateway between the spinal cord and this hindbrain structure. The spinal cord inputs provide information about the body's position in space, and these inputs from the pons relay information from the cerebral cortex, specifying the goals of intended movements. This allows us to plan voluntary movements in advance. And the cerebellum compares these types of information and then calculates the sequences of muscle contractions that are required to achieve each movement goal. Damage to the cerebellum thus can result in uncoordinated and inaccurate movements. As I've mentioned before, if you perhaps had a tumor in your cerebellum, this could lead to imbalance, and it would mess someone's coordination. Make sure you guys are practicing labeling on your neuroanatomy worksheet. You will need to label a sagittal section of the brain on your next exam. And you need to know all the structures we just reviewed, including the thalamus shown here in purple, the hypothalamus sitting below in turquoise. Later, we're going to teach you more about the pituitary gland that sits right below the hypothalamus. We're going to discuss these other structures from the midbrain, such as the tegmentum, and then the tectum, which it consists of the superior and inferior colliculi. We're also going to talk more about the pineal gland and its role in melatonin secretion. And then, of course, the pons and the medulla. And then don't forget that we'll also need to label the cerebellum. Make sure you also study the ventricles and where they're located. Remember that the ventricles are these important holes in the brain that contain the cerebral spinal fluid, also known as CSF. In this image below, it's easier to see the two pairs of lateral ventricles that sit beneath the overlying cortex. Here we have the third ventricle near the thalamus, the cerebral aqueduct that also plays an important role in CSF distribution, the fourth ventricle that sits in the hindbrain in between the pons and medulla and the cerebellum, and then the spinal canal, which is going to help provide CSF that's going down into the spinal cord. Make sure you review these key terms on everything we've talked about regarding the central nervous system and the peripheral nervous system, as this will be important for your next exam. Now we're quickly going to talk about brain imaging and brainwave recording. This allows us to look at different areas of the brain. First, we're going to talk about a CT scan, which is also known as a CAT scan. This is a non-invasive diagnostic imaging procedure, and the brain CT scans can provide us with detailed information about tissue and structures in the brain. That's better than a standard X-ray of the head. It's going to provide more data related to potential injuries or diseases of the brain. For example, a CT scan could be used to assess for concussions, intracranial bleeding, structure abnormalities like hydrocephalus or infections, lesions or injuries to the brain. So essentially, it's like a more detailed version of an X-ray. Now, an angiogram is a little more complicated, but this is a really cool process. It's an X-ray of the blood vessels. So see here, we can look at the blood vessels, and it's used to look for changes in the blood vessels. For example, ballooning of a blood vessel when it starts inflating is called an aneurysm. It's really dangerous if an aneurysm bursts in the brain, which could lead to blood in the tissue, which we know can induce excitotoxicity and other really scary consequences. So this could help somebody determine if they have a brain aneurysm. It could also help us look at narrowing of blood vessel and blockages in the brain, in the blood vessel specifically. We've talked about how lipids can build up in the blood vessels, and that could cause a blockage that could lead to a stroke. For an angiogram, a doctor is going to have to find a large blood vessel and inject it with a contrast dye. And so this contrast dye actually allows us to see the blood vessels in this X-ray image. And this can help us determine those things that I talked about earlier. An MRI, which is known as magnetic resonance imaging, is an imaging test that's used with powerful magnets and radio waves to create these really clean images of the brain. It allows us to see structures very easily. As you can see, we can point out areas we talked about today, like the cerebellum, and we can look at these important midbrain structures. We can see the corpus callosum right here. That is the main gateway for connecting the two hemispheres. And so essentially, this can allow doctors to look for tumors and aneurysms and other odd abnormalities in different areas of the brain. Another test that's used by neurologists is called positron emission tomography. This is also known as a PET scan. You may have heard of this before, but it's a type of nuclear scan. It can actually measure metabolic activity of neurons and other types of brain cells. And PET is usually a combination of both nuclear medicine and biochemical analysis. So, this is usually used for patients that maybe have brain or heart conditions or maybe even brain cancer. But PET scans specifically use a tiny amount of a radioactive substance, and this is used during the procedure to assist in the examination of metabolic changes in tissues specifically. It help us look at physiology. As you can see here, when the patient's eyes are closed, we don't see activity in this area of the brain. However, visual stimulation like white light can increase activity, and a more complex scene that is provided to the patient is going to increase even further visual stimulation and activity. So thus, this could be important for looking at metabolic changes in the brain specifically. A very common procedure that is used to look at abnormalities in the brain includes the EEG, which you may have heard of, and you may have even had an EEG performed on you before. But this can test and detect for abnormalities in our brain waves, which is the electrical activity of our brain. We've talked so much about electrical synapses in this class, so this can actually detect those abnormalities of those electrical synapses. And during the procedure, there are electrodes that are actually placed onto the scalp. This consists of these small metal discs with thin wires on the entire scalp. The electrodes can detect any tiny electrical changes that result from the activity of your neurons, and these charges are amplified and appear in graph form for physicians to see. And as you can see here, this is a great way for physicians to assess and evaluate brain disorders like epilepsy. So when epilepsy is present, seizure activity is going to appear as rapid spiking waves on an EEG, compared to a partial seizure where we may not see that drastic effect in the brain waves specifically.